Okay, welcome, my dear friends. This is Dr. Padala Tirupati, Assistant Professor, Department of Botany, SRR Government Arts and Science College, Autonomous, Karimnagar. We are organizing this national level free online coaching for all competitive exams of plant sciences. In this regard, today's our eminent resource person, Dr. Rani Kommidi Madam Garu, Head Department of Botany, Assistant Professor, Government Degree College, Siddipet. Madam previously explained about the plant embryology, at the same time paleontology also. Very crystal clear. Madam has presented all the classes during the extreme conditions, even though during night time also. So even though Madam has given her valuable time for the sake of the young aspirants' interest and also the people, those who are related to or regarding to the remote areas and village background, so they will get benefited during this online class and the platform, Zoom platform, online platform. So that's why Dr. Rani Madam Garu has come together and to give her valuable time to explain the most important area in plant physiology, that is nitrogen metabolism. So it is most important topic not only in the plant physiology, it is very close related or associated with the bacterial symbiotic association or the nutrition bacteria and also the bacterial association with the root nodules. And also it is very important in the mineral nutrition, that is the macro element or macronutrient of the nitrogen, the nitrogen physiological function or the impacts related to the deficiency of the nitrogen. So due to this reason, Madam has chosen this topic today. So we would like to invite Dr. Rani Madam Garu, Committee, Head Department of Botany, GDC Siddipet Autonomous. Madam, please welcome and start your session, Madam.
Okay, sir. Thank you, sir. Thank you for your kind words. And very good evening to my young aspirants. So due to lack of time, we are really taken some important topics on the plant physiology. So on that account today, we are going to discuss about the nitrogen metabolisms. So this is also very important, and every year 1 or 2 questions come from this topic only. So just wait. The slides are not moving. Wait.
Madam, please, you may comes out from the slide.
Now, okay, sir.
Slide show నుంచి మీరు బయటకు వస్తే ఒక్కొక్క స్లైడ్ ఈజీగా మూవ్ అవుతుంది. ఇట్లైనా పర్లేదు మీరు.
means wait. Slide show లోనే ఉంది, sir. Okay. So today we are going to discuss about the nitrogen metabolisms. So presentation on especially nitrogen fixation mechanism in the legumes or Fabaceae family. So just wait. This is the nitrogen bacteria, or just we are moving from the first. Okay. So nitrogen fixation in the legume families. So tomorrow we'll discuss about that. What is the basic concept of nitrogen and what is the essential of the nitrogen and nitrogen cycle. So on that account today, the nitrogen fixation mechanism in the legumes. So this is the nitrogen fixing bacteria. It's absorbed nitrogen gas from the air and it fixes into a form which the plant can use to build the protein. So we know that nitrogen in the atmosphere that is found in gaseous form, and it is measures about that 78.09% in the volume. So in our atmosphere, the nitrogen 78.09 or 78 point something. So that is in the volume. It is found in gaseous form in the atmosphere. So this elemental nitrogen that is not available to the higher plants, but certain prokaryotes like blue-green algae and some of the bacteria, they can utilize it. So however, some higher plants, they can utilize nitrogen only in different forms of the combinations. So like nitrogen, nitrate nitrogen, ammonia nitrogen, organic nitrogen, molecular nitrogen. So these all are the different combinations. They are available. So the higher plants can utilize these different forms only. So there are the nitrogenous, ammonium nitrite, nitrate, organic nitrogen. So these all are some different combinations only the higher plants can use it in their metabolic reactions. So in that, some of the blue-green algae or prokaryotes, they are also helpful for that nitrogen fixing bacteria. So they can absorb the nitrogen gas from the air and it will fix it into a form which the plant can use to build their protein. So they can change nitrogen gas into ammonium compounds like this is the root nodule, that bacteria, root nodule full of the bacteria. So the rhizobium bacteria, the main example of that rhizobium bacteria, the first rhizobia were isolated from root nodules by M. Beisering. Okay, so who will isolate that bacteria? The rhizobium bacteria are first time isolated from the root nodule by Beisering. So, and it's shown to have the ability to reinfect their legume hosts and it to fix nitrogen N2 form. So, molecular nitrogen in the symbiosis way. So, that's why this bacteria is the Rhizobium leguminosarum. This bacteria, Rhizobium bacteria, and especially Rhizobium leguminosarum it is a species of the gram-negative bacteria. The characteristics of bacteria is gram-negative bacteria, aerobic bacteria, and the size or shape. So it's look like a rod-shaped bacteria. It is found in soil only. Okay. So the Rhizobium bacteria is gram-negative, aerobic bacteria, rod-shaped bacteria, and it is found in soil and which causes formation of the root nodules. Okay. So on some, but not all types of the field peas, some Fabaceae, like we know that. So some of the legumes, we call that majority are the legumes. So the types of the field pea, lentils, kidney beans, and some of the clovers. So these all are just looking like a root nodules. They can form the root nodules in their root system. So that will help from this rhizobium bacteria only. Okay. So this bacteria can infect the roots of the leguminous and it leading to the formation of the lumps or nodules where the nitrogen fixation takes place in their area. So about 90% of the legume species, they can become the nodulated. So this is the completely the structure of the characteristics and structure of the rhizobium. Look like a gram-negative bacteria, aerobic, rod-shaped, and it's completely found in the soil only. It will infect or it causes the formation of the root nodules. So the nodule formation, generally the nitrogen fixation, it will takes place by two types of the microorganisms. We know that. So asymbiotic or free living or symbiotic that live in the association with other plants. Okay. So in this symbiotic nitrogen fixers, the symbiotic nitrogen fixing systems are found in many vascular plants. So, and they are the some of the non-vascular plants also. A number of angiosperms, that means members of the family Leguminaceae. So as I told you, and some of the other families, and some are the gymnosperms and fens. So they are also associated with the various nitrogen fixing microorganisms. These vascular plants, they can serve as host to such microorganisms. So in that way, some of the symbiotic nitrogen fixing system, they are look like a other way. So they are also, especially the rhizobium, it's formed the nodule, so root nodule. The nodule formation takes place, rhizobia attracted to the root. So this is the completely it will found in soil only. So that's why the rhizobia, they are attracted to the root system, and the rhizobia attached to the root hairs, and it is cause that root hairing curling. So the curling is just change it to the infection thread formation. So just to we are discussed one by one. First, the nodule formation is followed by this way. Rhizobia bacteria attracted to the root portion and rhizobium. So it will attach to the root hairs, and the root hair is modified as a curling due to the cell division, due to the growth of the rhizobia or functioning of the rhizobium. So the hair is look like a curling, and the infection thread formation, and the root cortical cell undergoing the divisions. So the rhizobia, they can invade cortical cells, and the nodule is look like a bulb-like structures or some lumps. Nodule grows, so it's look like a fully functional nodule. These all are changes is happen by this bacteria whenever attached to that root portion. So the leguminaceae or other plants, the rhizobia first attracted to the root, and it attached to the root hairs. So due to just it will secrete some enzymes. So due to that enzymes, it will attach to the root portion, root hairs. So the root hair is modified, curling. So infection thread is formation, and root cortical cell undergoing the division look like a complete reddish color. So some of the other pigments are formed. So the root cortical cell division, and rhizobia is invade the cortical cells only. So the nodule is look like a round shape. It will undergo the grow. So or look like a bulbil structure. So almost finally fully functional nodule is formed in the root portion. So this is the mechanism of nitrogen fixation in root nodules of the legumes. Okay. So in the legumes, look like a nitrogen fixing bacteria, rhizobium, that fix atmospheric nitrogen. Okay. So in this way, generally to understand the physiology and genetics of nitrogen fixation, the nitrogen fixation at the biochemical and chemical level is essential in this way. So these details on such levels have been look like studied by Postgate in 1971 and EADY. So these both are worked together on these metabolic actions. So an important enzyme like nitrogenase. So which enzyme, Nana? Nitrogenase enzyme is attributed to show the nitrogen fixation in the cell-free extract. So this is the complete soil. The plant is growing in the soil. So almost the bacteria is almost it is also grows in the soil only. So it will attract to the root portion, the nitrogen fixing. So molecular nitrogen can modified. ఇక్కడ నుంచి మనకి ఏంటి అంటే molecular nitrogen అనేది just it will modified as, so nitrogen fixing bacteria is rod shaped, and it's modified as some nitrogenase enzyme is modified as ammonia and nitrifying bacteria. So nitrate, so denitrifying bacteria, all will just it's called as a some nitrogen cycle. So this is also we'll discuss. It's look like a electron donor. So the glucose 6-phosphate is converted to the phosphogluconic acid, and in this, so this is completely So in this way, the enzyme, an important enzyme, nitrogenase is attributed to, they can show the nitrogen fixation in the cell-free extracts. So the enzyme system has been extracted from 16 microorganisms like Clostridium and Pasturinium and Bacillus, Klebsiella, Mycobacterium, Azotobacter. So these all are the, just the system has been extracted from 16 microorganisms. Like all these belongs to the some prokaryotic bacteria. So the Clostridium and Klebsiella, Mycobacterium, Azotobacter, Polymixa. So these all are helpful for that. So this enzyme look like they can including the 4 symbionts with leguminous. So this enzyme reduce the nitrogen to the ammonia. So, but no intermediate has been detected at the level of the hydrazine or diamine. So in this way, the reactions require ATP. ATP also are requiring, which is transformed to the ADP and orthophosphate, and they can reducing the reagent to the function as electron donor. So in this, especially the pyridoxine. Generally, pyridoxine or a flavodaxine. Pyridoxine, it will serve as an electron donor to nitrogenase, even in the aerobic nitrogen fixing systems like Azotobacter or some or the other legume nodules also they are involved. So in this way, some sodium dithionite or sodium hydrosulfide, it will react directly with the enzyme system, and that is used in the laboratory. So in this way, some magnesium is essentially required for the nitrogen function. So 2 mg ATP also is modified consuming in this way. So it will modified as ADP and IP. So the magnesium is essentially required for the nitrogen function. Here, some reduced pyridoxine are produced in the just phosphoclastic reaction. So this is the pyridoxine is flavodaxine. Pyridoxine or flavodaxine, and it's modified as a some with the help of the ATP, iron protein also formed in this way. So the reduced pyridoxine are produced in the phosphoclastic reaction, and in this way some of the acetyl CoA also formed. Just one slide is missing, I think. So acetyl CoA formed in the some first reaction. So the ATP generates via reactions according to the just to some the enzyme nitrogenase is made up of the 2 proteins. So in this way, one of molecular weight around they can 220,000 having molybdenums and non-heme iron and some of the other also formed in this reaction. A small protein of molecular weight about 70,000 having the only non-heme iron and some of the labile sulfide. So these all are some of the nitrogen fixing. The enzyme nitrogenase is characterized by metalloenzyme character. So it will extreme oxygen sensitivity and waste of the ATP also. So just in this way, now the question arises, how to aerobic nitrogen fixing microorganism? It will symbiotic system or it protect the functioning enzyme from the oxygen damage like O2 damage. So however, some postgate he described in 1975 4 methods to avoid the oxygen damage, like oxygen restricting mechanism. They can enable aerobic organisms to fix the nitrogen, like respiratory protection and some of the conformational protections or heterocyst formation or the root nodule as a compartment. So these all are the just to he described one by one. So the reduced iron protein combines with ATP in presence of magnesium. Second subunit is activated and reduces. It donates electrons to molecular nitrogen to the NH3 ammonia, and enzymes set free after complete reduction of the N2 to the NH3. So just to what is the nitrogen cycle? So one by one, in the nitrogen cycle, so this is the two basic types of the biogeochemical cycles. Biogeochemical cycles means some of the important elements, they can already, they are present in the soil, and some of the living organisms will uptaken, and they can, after dead and decay of the living organisms, some of the microorganisms will, they can almost complex organisms will modified as a simple, and they buried in the environment. So soil, living organism, microorgans, all will just the elements are looking like around of the biogeochemical cycles. So that is look like a gaseous cycles. So the sedimentary cycle, almost biogeochemical cycles are of three types. So in this, so the biogeochemical cycles, they can describe the pathways by which essential nutrients and chemical elements move from the, they can move from this soil to the living organism. Living organisms, they can need 30 to 40 elements for normal development, and the chemical elements or nutrients, they can flow from the non-living to the living and back to the non-living components, so of the ecosystem, in a more or less cyclic pathway. So this is known as the biogeochemical cycles. So it is look like a sedimentary, gaseous cycles and sedimentary cycle. Gaseous cycles are carbon cycle, nitrogen cycle and oxygen cycle. So in this, the nitrogen is the very important. Nitrogen is converted between its various chemical forms through following steps. So like nitrogen fixation, ammonic fixation, nitrification, denitrification, and fifth step is the assimilation. So this is the complete forms. The nitrogen cycle is following these steps. First one is the nitrogen fixation, ammonification, nitrification, denitrification, and assimilation. So the completely the nitrogen in the environment look like a 78.08% in the value. So it is look like a gaseous form. Okay. So this element nitrogen, it is not available to the higher plants. Some of the prokaryotes like blue-green algae and bacteria, they can utilize it. Some of the higher plants can utilize the nitrogen only in the different combined forms like nitrogen, nitrate nitrogen, ammonia nitrogen, organic nitrogen, and molecular nitrogen. So what is the importance of nitrogen? So nitrogen is the essential for life, as it is the part of nucleic acids, amino acids, proteins, enzymes, and other coenzymes like NAD, NADP, FAD. So these all are the coenzymes, and some of the cytochromosomes and vitamins, chlorophylls. So these all are, they have this nitrogen part and parcel of the their association with this nitrogen only. No doubt that nitrogen makes up the greatest part of the atmosphere, but green plants can use little or no atmospheric nitrogen like N2 form in their synthesis. So some of the green plants can use nitrogen, molecular nitrogen as ammonia or in the form of nitrates, and they are also obtained from the nitrogen fixing bacteria of soil, which can utilize the nitrogen directly in the synthesis of their own amino acids and proteins. So this is the essential constituent of plants and animals. The plants take up nitrogen either as nitrates or ammonia. So due to thunderstorms and lightning, the gaseous nitrogen is converted into the nitric oxide also. So what the process of the nitrogen cycle? Nitrogen, it's present in variety of forms. So the organic nitrogen, nitrous oxide, or inorganic nitrogen gas, So the process of the nitrogen cycle, it will transforms nitrogen from one form to the another. So this is the nitrogen cycle. The major reservoir of nitrogen is the atmosphere. So we know that 78.08 or 09, whatever it. So it is also important question in your examination point of view. So the major reservoir of nitrogen, it is in the atmosphere, look like a, it is the atmosphere is found in gaseous form and measures about 78.08 or 09 in volume. Okay, so various steps involved in this cycle. Nitrogen fixation. So it's look like atmospheric free nitrogen. Nitrogen fixation are of two types, non-biological fixation and biological fixation. So non-biological fixation. So the physical process or high energy fixation, it's look like a natural process or man-made process. So in the natural process like photochemical fixation, it is also in the other name. Natural means photochemical fixation and man-made is industrial fixation. Okay. So natural means thunderstorms or any other changes in the environment, that is the photochemical, and man-made just who is involved by the human beings only, like industrial fixation or any other chemical factories. So that is the photochemical fixation or natural fixation. So it is look like a dissociation of nitrogen molecule and its combination with oxygen and hydrogen of water provided by lightning is photochemical fixation. Okay, so look like the transformation of atmospheric gaseous nitrogen, look like so that molecule and its combination with oxygen and hydrogen of water provided by lightning is the high energy is required for this process, and it is provided by cosmic radiation and meteorite trails, like మనకి భూకంపాలు వచ్చడం రావడము తర్వాత తుఫాన్ ఏమంటారు థండర్ స్టార్మ్స్. ఓకే, సో దే ప్రొవైడెడ్ బై కాస్మిక్ రేడియేషన్ అండ్ meteorite trails. And industrial fixation or man-made fixation, so the fertilizers in various industries look like a urea, so whatever it combinations, NPK, nitrogen, phosphorus and potassium, so that is also fertilizers in various industries. Most chemical fertilizers are nitrates or ammonia, so for agricultural field to yield crop and increase the soil fertility also. Okay, so this is the, and next one is the biological fixation. So in the biological fixation, it is very, very important. Generally, the conversion of atmospheric dinitrogen, like molecular nitrogen, N2 form. Okay, the conversion of atmospheric dinitrogen into a chemical form which plants and microbes can use for growth is called as the nitrogen fixation or biological nitrogen fixation. Biological nitrogen fixation means the conversion of the atmospheric nitrogen like N2 form into a chemical form, so which plants and microbes can use for their growth. So that is called with the help of microbes kabatti, so with the help of that it is called as the biological nitrogen fixation. So because it is and second is organisms, they can living in the soil independent of host plants like azotobacter or clostridium. And third one, a group of photosynthetic bacteria and blue-green algae. So this is prokaryotic bacteria also helpful for that biological nitrogen fixation. Okay, so that is the nitrogen is fixed in the form of ammonia and ammonium salts or nitrates. So the nitrogen fixing organisms are called diazotrophs. So this process is symbiotic and non-symbiotic nitrogen fixation. So in the symbiotic nitrogen fixation, so it carried out by microorganisms, the symbiotic nitrogen fixation only. So the rhizobium that lives in the root nodules of leguminous plants. Okay, so that means the symbiotic nitrogen fixing systems are found in many vascular plants and non-vascular plants also. So this is the non-symbiotic nitrogen fixation, like asymbiotic also. So they are again of 3 types, carried out by microorganisms living free in the soil only. So for example, bacteria, Bacillus bacteria, Clostridium, and some of the aerobic forms like Azotobacter. And third one is the blue-green algae. So that is the cyanobacteria like Nasta, Cannabina, Oscillatoria. These all are the non-symbiotic or asymbiotic. That means, so the nitrogen fixation takes place by 2 types of microorganisms, asymbiotic or free-living or non-symbiotic. The second one is the symbiotic. Symbiotic means that live in association with other plants is the symbiotic. Okay. Asymbiotic means, so they are free living nitrogen fixers. The asymbiotic or non-symbiotics are the free living nitrogen fixers. So these asymbiotic are again of the 3 types. So like aerobic bacteria, anaerobic bacteria, and blue-green bacteria. Okay. So aerobic bacteria is example Azotobacter. Anaerobic is the Bacillus Clostridium. So that is, and other one is the blue-green algae, like cyanobacteria, Nostoc, and Oscillatoria. Okay. So this is generally the free-living anaerobic bacteria occur in many soils, but they furnish significantly to the soil nitrogen under special conditions like abundant plant tissues and high water content. Okay, so just to almost majority of you know that in blue-green algae, they can have the heterocyst. So the heterocyst, they can fix the nitrogen. Okay, so the blue-green algae have important contribution in rice fields especially. The presence of heterocyst is an indicative of the nitrogen fixing ability in the free living forms. So in lichens, the formation of the heterocyst is not a prerequisite for the nitrogen fixation. That is the non-symbiotic or asymbiotic or free living. Okay. So look like fixation, and second one is the ammonification. Okay. So in the nitrogen cycle, first step is the nitrogen fixation and second step is the ammonification. So the ammonification, it's look like a organic matter converted to the ammonia. So the heterotrophic bacteria like actinomycetes and fungi, and they are also look like a organic nitrogen inorganic form as ammonia. In this way, some of the energy releasing reactions are involved in this way. So that is the ammonification. So simply, ammonification means the nitrogen, molecular nitrogen can changed as the ammonia is known as the ammonification. Okay. So the nitrogen in the soil is also found in reduced form in NH4 plus. Okay. So NH4 plus is known as the ammonium ion. So NH4 is absorbed by the roots of higher plants and they can participate directly in the metabolism. Okay. So that is the ammonification. Ammonification is look like, so it's the source of ammonia in soil is artificial introduction as a chemical fertilizer or microbial action. So they are depends upon dead and decaying organisms also. Some of the paddy plants, they are known to absorb the ammonia as nitrogen form so rapidly. అక్కడ ఏంటి అని అంటే some ammonification means molecular nitrogen can modified as a ammonia is known as the ammonification. Okay, so the ammonification, organic matter converted to the ammonia, NH3 or NH4 plus. So in this, some of the heterotrophic bacteria are helpful for that. So that is the completely like heterotrophic bacteria, actinomycetes, and fungi is also helpful for this reaction. So some of the organic nitrogen is inorganic forms as ammonia. So that is also just changes in this ammonification steps, the energy releasing reactions also formed in this way. And the next one is the nitrification. So nitrification, so nitrification, ammonia or ammonium salts is nitrites and nitrates. Okay, so in this process is known as the nitrification. NH3 cannot be utilized directly. So in the NH3, it cannot be utilized directly in this step. So that is the nitrification. Ammonia or ammonium salts is modified as a nitrites and nitrates. NH3 plus, sorry, NO3 minus and NO2 minus. NO3 minus or NO2 minus. So this process is known as the nitrification. So in this, some of the bacteria like nitrosomonas or nitrosococcus will helpful for this step. So that is the nitrification, and NH3 cannot be utilized directly. So this is the denitrification. Bacteria and fungi convert the nitrites, so nitrous oxide, NO2 minus, NO3 minus, and NO minus. So that is nitrous oxide. This is known as the denitrification. So the denitrification means conversion of nitrates to the gaseous nitrogen gas, like NO3 nitrate, so NO2 nitrite, and NO2 nitrous oxide. So look like NO nitric oxide, and last step is the nitrogen gas, molecular nitrogen or dinitrogen. So that is the changes are occurring in this step. That is the denitrification with the help of some other pseudomonas. Denitrifying bacteria are the pseudomonas and thiobacillus. Okay, so that is the way bacteria and fungi convert the nitrates and nitrous oxide. So the denitrification, conversion of nitrates to the gaseous nitrogen to, so that means nitrite to nitrous oxide, NO2 minus to the NO2, and NO2 is modified as the NO nitric oxide, and finally, so as usual, the molecular nitrogen or dinitrogen is formed in this. So that is the denitrogen. Okay, so in this, in the nitrification, denitrification, so in this process, the ammonia is converted into nitrate by the presence of bacteria in the soil. So the nitrates are formed by the oxidation of the ammonia with the help of nitrosomonas bacterium species. So later, the produced nitrates are converted into nitrates by nitrobacter. So this conversion is very important as ammonia gas is toxic for that plants. So the reaction means in the second step, like nitrification step. So 2NH4 plus 3O gives rise to NO2. So that is missed like, and denitrification. So in this, some bacteria, fungi is converted to the nitrates to nitrites and nitrous oxide. So the denitrifying bacteria are Pseudomonas and Thiobacillus. So the conversion of nitrates to the gaseous nitrogen gas are formed in this step. And next one is the assimilation, like some of the primary producers, like plants. So plants take in the nitrogen compounds from the soil with the help of their roots only. Okay, so which are available in the form of ammonia, nitrate ions, or ammonium ions, and all are used in the formation of the plant and animal proteins. So this way it enters the food web when the primary consumers are eat the plants. So that is known as the assimilation. So the principal source of nitrogen for plants look like a nitrate ions and water. So the nitrate assimilation, it absorbs nitrates for the synthesis of the proteins means. So in this way, some of the primary producers like plants, so take in the nitrogen compounds from the soil with the help of their roots. So the roots can uptake in the some of the elements, so which are available in the form of ammonia, nitrate ions and nitrate ions. So they all are used in the formation of the plant and animal proteins. So this way it's look like enters the food web when the primary consumers are eat the plants. This is the complete picture of this nitrogen cycle. So in the nitrogen cycle, look like a completely stages of nitrogen cycle is the nitrogen fixation. So means the process of nitrogen cycle consists of the following steps: nitrogen fixation, nitrification and denitrification, so ammonification, assimilation. So this process take place in several stages and they are look like a, so nitrogen fixation, it is the initial step of the nitrogen cycle. Here some of the atmospheric nitrogen which is primarily available is an inert form. So that is converted into the usable form like ammonia, NH3. So during this process of nitrogen fixation, the inert form of nitrogen gas is deposited into soils from the atmosphere and surface waters. So mainly through precipitation. Some or later, the nitrogen undergoes a set of changes in which two nitrogen atoms get separated and combine with hydrogen to form the ammonia. So NH4 plus. So the entire process of nitrogen fixation is completely by symbiotic bacteria, which are known as diazotrophs. So like Azotobacter and Rhizobium. So they also have a major role in this process. This bacteria consists of a nitrogenous enzyme, which has the capability to combine gaseous nitrogen with hydrogen to form the ammonia. So in this, the nitrogen fixation can occur either by the atmospheric fixation, which involves lightening, or industrial fixation by manufacturing ammonia under high temperature and pressure conditions. So this can also be fixed through man-made processes, primarily industrial processes that create ammonia and nitrogen rich fertilizers. So that is the some nitrogen fixation. Like the types of nitrogen fixation: atmospheric fixation, industrial nitrogen fixation and biological nitrogen fixation. So all will discussed. So nitrification and ammonification, denitrification, assimilation. So generally, what is the importance of nitrogen cycle? So the importance of nitrogen cycle are, it helps plants to synthesis chlorophyll from the nitrogen compounds. Okay. So it helps plants to synthesis chlorophyll from the nitrogen compounds. And second one is it helps in converting inert nitrogen gas into a usable form for the plants through the biochemical process. So in this process of ammonification, the bacteria help in decomposing the animal and plant matter. Okay, so which indirectly helps to clean up the environment also. And the nitrates, they are released into the soil, which helps in enriching the soil with necessary nutrients required for the cultivation also. So finally, the nitrogen is an integral. So it is look like a integral component of the cell, and it forms many crucial compounds and important bio molecules also. So the nitrogen is also cycled by human activities such as combustion of fuels and the use of the nitrogen fertilizers. So these processes increase the levels of nitrogen containing compounds. So the fertilizers containing nitrogen are washed away in lakes and rivers and resulting in the eutrophication purpose. So this is the completely nitrogen cycle. Just today we are discussed only the nitrogen cycle, and tomorrow, if possible, we are going to discuss about the clearly mechanism of the biological nitrogen fixation. So it's very, very important, and just to that is the way. So this is also some complete steps are involved. Plants and nitrates, nitrites, ammonia, decomposers. So all will assembled in the atmosphere. So it's look like a cycling model. So this is the nitrogen cycle. And the conclusion, nitrogen is an important component. So this conversion is a cyclic process. The process of nitrogen cycle transforms nitrogen from one form to the another form. So with this, the only what is the importance of nitrogen and the nitrogen biogeochemical cycles, only today the nitrogen cycle we'll discuss it. So tomorrow completely the symbiotic, mainly biological nitrogen fixation is very, very important. The mechanism, tomorrow we'll discuss that. So with this, I over to Tirupati sir. Hello sir.
Thank you very much, madam, Dr. Rani Madam Garu. During busy schedule, you are also you giving your valuable time for our students, young aspirants. So thank you very much, madam. Tomorrow we will meet once again. Okay. Thank you.
Thank you, sir. Okay, right.